So here we go. Welcome back, folks. Now where we are in your checklist, we are on page 8. And what we're going to talk about right now is the cell structure. Now, the thing is, again, for lab purposes only, we are only going to have you identify the structures of the cell. In lecture, we will be more specific on what the actual cell and the organelles, the little structures that make up the cell, what it actually does. So for our purposes, we have this wonderful cell model right here that we you'll be able to, that we can put pictures up on a test possibly. But there is also a good cell model picture in the APR. So again, if you get a chance, make sure you're looking at the APR picture of the cell as well. So when we first start off, if we are letter A, the cell or the plasma membrane, that's just going to be the outer border of the cell. And again, what does the cell membrane do? That separates the internal components of the cell versus the rest of the body. And that's all that function is. Now the next structure, as we come down, is the nucleus. And the nucleus is this large circular structure right here, right in the middle of the cell. This nucleus is actually what controls the functions of the cell. As we come down and we keep moving down the checklist, the nucleolus is this small circular organelle right here that sits within the nucleus. And again, we will explain more of its function in lecture. So again, this is another very big point, folks. Know the difference between nucleus and nucleolus. That again, very first-time AP students, very similar sounding, very easy to confuse. The nucleus is the large organelle right here, and what sits right in the middle of it, that is the nucleolus. As we come down, the nuclear envelope. We don't have the nuclear envelope or membrane, but it's a membrane that covers the nucleus. And again, what it does, it contains a large number of pores in it, and that allows substances to move in and out of the nucleus very easily. And we'll talk about its importance in lecture. The next term is chromatin. What we need you to know, you really can't see chromatin on this model, so we're not really going to discuss that much. But just make the note while you're looking at this video, chromatin is our DNA. And when our cells are not actively dividing, our DNA becomes stretched out into long, fine strands of what we call chromatin. When our cells start to divide, the chromatin condenses down into visible chromosomes for us to see. But again, chromosomes are still made up of DNA. It's just a matter of how the DNA is packaged. And chromatin is shown, and chromatin is found within our nucleus. As we move down, the next one is cytoplasm. The cytoplasm is the internal component of the cell. And then if you look in your checklist, it includes the systole. The systole is just quite simply the fluid that is found in our cells. And it's also the fluid is what suspends all of these little structures in our cell called organelles. So the entire inside component of the cell is called the cytoplasm. And the cytoplasm is made up of the cytosol, which is the fluid component, and of the organelles, which are suspended in that fluid. Continuing down the checklist, you have the rough endoplasmic reticulum. The rough endoplasmic reticulum are these structures right here. It's these orange structures with all these little purple dots on them. That is the rough endoplasmic reticulum. Smooth endoplasmic reticulum is not found on this cell model, but it is found on your APR picture of the cell. The big difference between rough and smooth endoplasmic reticulum is rough endoplasmic reticulum contains ribosomes. Ribosomes are organelles that produce protein for our body. And if we come up real close, you can see all these little purple dots right here that are attached to these orange structures. This would be the rough endoplasmic reticulum. Could we have ribosomes attached to the endoplasmic reticulum's membrane? On this, there's no smooth endoplasmic reticulum. Free ribosomes. Free ribosomes would be found out in the systole by themselves, and on this one it's very difficult. In fact, there are none on here. But again, if you had the ribosomes freely circulating within the fluid portion of the cell, that would be considered to be a free ribosome. And again, we will get into that discussion on the importance of our fixed ribosomes versus our free ribosomes. But again, our fixed ribosomes are quite simply attached to the rough endoplasmic reticulum. As we come on down, the next structure is the Golgi apparatus. The Golgi apparatus is this structure right here, and it looks like a set of dinner dishes that are stacked on top of one another. And actually what it is, it's a membranous structure. So again, we will talk about functionality in lecture. As we come down, the lysosome, this little blue structure right here, that is the lysosome. What its job is to circulate around the cell to keep it free of waste and debris. The mitochondria. The mitochondria are these kidney bean-shaped structures that are found throughout the cell. And mitochondria's function, quite simply, is it produces ATP, which provides us energy for our cells. And the last one is a centriole. Now this model right here is only showing one centriole. All the cells that contain centrioles contain two centrioles. And what the centrioles are important for, the centrioles are essential for cell division to occur. And again, I believe the APR picture in APR of the cell contains two centrioles, but the cell models only contain one.